Oops. Welcome to everybody. Very happy that everybody is joining for our third iteration of the Cluster of Research Excellent Advanced Infectious Disease Training of our European African Network. And while we're waiting for Andy to come back on, first a couple of housekeeping announcements. If I can move to the next slide. So I'll be chairing the session. My name is Taco Koy. I work as an assistant professor at the Radboud UMC. And today we have two talks by Andy Waters from the University of Glasgow and another talk from the University of Glasgow by Mariana Ferreira Silva. And I'll introduce them later when we get to the talks. Please keep all your mics muted during the presentations. Everyone is welcome to write their questions in the Q&A box. I'd like to stress that this is a respectful and inclusive space. No discriminatory and/or harassing behavior will be tolerated. The meeting is being recorded automatically, so recording has already started, so including our technical hiccups, and the recordings will be shared with all the participants. As your feedback is very important also for the further development of the cluster, please fill out the post-attendee survey at the end of the webinar. This will only take you a few minutes. And I'm wondering, Andy is back? Yeah.
I should be able to share, but of course I haven't been able to check that yet.
Well, we'll be in for a surprise to see if it works. I'll stop sharing and start with my introduction while you try and get the slides up. Not that Andy needs much of an introduction, really. I think most people will be very familiar with Andy and his work. Yeah, sharing seems to work.
Okay, I'm glad you can see it because I can't. Right, okay.
Yeah, and we see a presentation as well.
And is it full screen?
And it's full screen now. Wonderful.
Well, technology.
Wow.
I did actually find something to click on, which clearly did the trick.
It only took us 20 minutes.
Oh, good grief, did it? Okay. Right, well, let's crack on. Good afternoon, everybody. Thank you, Taco, for the introduction. I'm going to rush through some work that we've been doing in collaboration with Thierry Diagana at the Novartis Institute for Neglected Tropical Disease. And it involves drug resistance. And as the first slide demonstrates, taken from a review from David Fiddick's group, that drug resistance is inevitable. Every drug that we've launched on in combating malaria, then that's at some point or other, sometimes very, very quickly, even in trial stages, then resistance has been observed. So what we want to look at is, rather than bore you with the malaria life cycle, I know it's not boring, but I know you know all about it, is that WHO have targeted transmission blockade as a priority area for the development of intervention measures and drugs that might prevent the formation of gametocytes or prevent fertile gametes being formed. Any of those phases have the potential to be valuable in this enterprise. So the work I'm going to present, the lead people have been Cam2, Jonathan, Nelson, and Nisha, generating all of the data. If then we can actually prevent something that makes gametocyte formation or any of those forms that are in the bracket, then of course we may well have something that would be useful in the overall combating of malaria. So this is ganaplacide. It's a drug that's been developed by the groups of Thierry Diagana at Novartis. And it's an imidazole piperazine, which is a very promising chemical space for combating a lot of different syndromes, not just infectious disease. This particular molecule, CAF156, now renamed as ganaplacide, is extremely promising. It has completed its phase 3 clinical trials. It's in combination with a second drug, lumefantrine. The answer to the problem of resistance is that we actually use combination chemotherapy. Has been pioneered with artemisinin. Although unfortunately we deployed it too late with artemisinin because we'd used it as a monotherapy in the first place, allowing resistance to be established. What we're looking for is something that will block that, and ganaplacide does do that. It kills all blood stages and prevents transmission. So at one level, it's already doing the job it should do, and certainly, ideally, if it's going to kill gametocytes, it might as well kill asexuals as well, because then you're getting double the bang for your buck. So that then presents the onward transmission of the parasite. However, resistance has been demonstrated against these drugs, and this article made us prick up our ears in our group here when it was published in 2016, demonstrating that UDP galactose and acetyl-CoA transporter genes were plasmodium multi-drug resistance genes, and one of the drugs that these conferred resistance to was indeed ganaplacide. There's a third gene which has also been selected in Plasmodium falciparum. One of the interesting things about this study was that they not only did the full gamut of generating resistance, sequencing the parasites, looking at the point mutations, and then going back and doing reverse genetics to demonstrate that those observed mutations did indeed give rise to resistance to the specific chemical entities. But they did this both in P. falciparum and in Plasmodium berghei. And both selection regimes, which are different, of course, because falciparum is kept in culture and berghei is largely kept in vivo, and certainly for the generation of resistance would be kept in vivo. The acetyl-CoA transferase mutations were observed in both species, and in fact, that's the only gene that's been selected in vivo for and implicated in the resistance to ganaplacide. And multiple mutations, the asterisks that are shown at the bottom here, here and here, and I think there's one there. I can't see on my screen, unfortunately, because of the pictures. That demonstrates that you can get stop mutations, or you can get nonsense mutations, or you can get and the rest are then just amino acid replacements. So only the acetyl-CoA transmutations have been selected in vivo. And then you compare the genes in the two species, then you can see from the number of asterisks that there's a huge level of identity there. Comparing the falciparum and the berghei, falciparum on the top. The boxes show you where the different mutations have occurred, and none of the mutations are identical, but some of them are closely juxtaposed, and all of them take place in regions which are highly conserved. That conservation is shown here when you look at the predicted secondary structure of the protein, and the little coloured balls show you where the mutations have arisen. And you can see that the overall structure is extremely similar. They're both multipass membrane proteins located in the endoplasmic reticulum of the parasite, which is shown for falciparum on the left and berghei on the right. These mutations then do give rise to resistances, as shown here. So what does resistance look like? Well, we've only got one ACT mutation here. This is taken from the Elizabeth Winsler paper, showing that resistance is generated to CAF179. This graph here shows that, as always, or is generally true, a resistance mutation confers a selective disadvantage in terms of growth and competition against the wild-type parasite when there's in the absence of drugs. So the selective advantage is only conferred when in the presence of the drug. So the declining lines here demonstrate that the wild-type parasite does indeed outgrow the mutant, and that's shown here as well. in the presence and absence of drug. So we were interested because Nisha had already knocked out the ACT gene for completely different reasons in our own research. So we went on to test whether or not this, and Nelson tested this to see whether or not it was indeed resistant. And the knockout indeed confers sevenfold more resistance to ganaplacide compared with the wild type parasite. And again, we see a growth rate difference so that the wild type parasite in the red in vivo, when put into competition with the resistant form of the parasite, the knockout, then the wild type quite rapidly overgrows the mutant. So our mutants were, we tested them whether or not mutants were that were engineered to reproduce the mutations that had been observed in Plasmodium berghei in the Winslow study from the in vivo selection in Plasmodium berghei. We then had a look to see whether or not those confer resistance, and you can see M1 and M2. These are just two selected mutations. Both give rise to significant resistance to the drug. And remain sensitive to artemisinin. And they also have a growth deficiency when compared with wild type parasites and are unable to effectively compete for growth. So what about gametocytes? Do they kill them? So we used our long-standing reporter parasite line, which gives red females and green males, which we can then use here on a fax screen to look for the male gametocytes expressing GFP and the female gametocytes expressing red fluorescent protein. And we can then have introduce these mutations into that background and have a look to see what the effect is. So you can see here that in an artemisinin-resistant parasite that we also engineered, then we still get male and female gametocytes. However, growing the parasite in vivo with the two mutations that we were testing, you can see that there's virtually no female gametocytes whatsoever. So that was extremely interesting to us. And when we tried to transmit these parasites, then neither of the mutants were able to transmit through the mosquito, and indeed they fail to mate and make productive ookinase, which is something that our model allows you to do in vitro. So the drug is extremely good at preventing transmission, but resistance also, in the absence of drug, generates parasites which are unable to complete gametocytogenesis and give rise to a productive infection in the mosquito. So that seemed like possibly the end of that story. But then one of the things that we can do with Plasmodium berghei, and we do that in order to synchronize our parasites, is we take the blood from the infected animal and pop that into culture. The schizonts will mature, and any gametocytes in there would also have the opportunity to mature. So we decided to test whether or not that was the case. So you can see when we look at it in vivo, you see again some male gametocytes and an absence of female gametocytes. Then when we let those mature, either the knockout or either of the two mutants, then the parasites progress to full maturity. And indeed, when we put those into the in vitro cultures that we can perform, you see at the top here, the wild type parasite forming the typical banana-shaped ookinate, highlighted by a surface marker, and the two mutants, M1, M2, and the knockout, are all capable then of making ookinates. And we're in the process now, after a lengthy delay, of trying to assess whether or not through membrane feedings we can actually transmit these mutant parasites. So what's the nature of the arrest? So Nelson then did an RNA-seq experiment where he infected an animal with two of the mutants, M1 and M2 that you see here. What he did was allow the mouse to get to a certain parasitemia, bled the mouse, processed half of the blood immediately for RNA-seq, and then matured the rest of it for 14 hours to get mature gametocytes, and then that allowed us then to do a simple comparison. And we looked at the differential genes expressed compared bleed out compared with after culture. then comparing them with wild type, you can see that the large number of genes that were underexpressed in the mutant at bleed out, but that the extended culture and the apparent maturation of the gametocytes was completely reproduced in terms of the gene expression, with a minimum of 2 genes which seem to be still differentially expressed after maturation in one of the mutants. When we mapped these 225 genes to the single cell progression that's produced in Matt Marty's lab and helped by Thomas Otto, then as we'd seen with the expression of the red fluorescent protein in the female gametocyte, the vast majority of those genes were in fact expressed in the female, but 2 of them were indeed expressed in the male. And we've gone on to show that with crosses with competent gametocytes, that both the male and the female are incompetent. The male will actually not exflagellate. So we took several of those genes, and Jonathan's joined the story at this point. And he examined the effect of knocking out some of these genes and the ones he could knock out. And there were the 2 genes, sorry, that were male expressed and differentially expressed in the RNA-seq experiment. He knocked out the NEC3 NEMA-related kinase, which is a gene associated with DNA replication. And indeed, the male parasites are defective in that. Females are perfectly okay. They can't exflagellate and they don't replicate their DNA effectively. So it looks like the phenotype of the arrested gametocytes in the blood stage is both male and female, and even that 1 gene in the male line recapitulates the phenotype that we observed. So we want to know even more about that. So we're making use now of the gametocyte inducible system that's been developed by Robin Kent and Katie Hughes in our lab, and with help initially from great deal of input initially from Kazia Madrinska. Effectively what we have now is a line that we can flip a promoter so that it drives the master regulator of gametocytogenesis, the conserved master regulator, AP2G, so that when we add rapamycin experimentally, we can switch from driving the expression of the selectable marker through to driving the expression of AP2G. That allows us then to have a synchronous induction of gametocytogenesis, which we can then follow. We have 2 different selectable markers, either the original line using a drug selectable one, or a fluorescent selectable one, which Katie has recently developed and deployed and is now published. Then you can see that when we do that switch, that most of the cells actually go on to then become male and female gametocytes, and typically not far off the same ratio that you would expect to see in a wild-type parasite. So Jonathan devised this really interesting experiment comparing induced and uninduced parasites, and then comparing in vivo with in vitro matured parasites. And for the in vivo parasites, he's injected induced parasites at these different time points after induction, so that they and injected them into the mouse, so that one mouse might receive as many as 3 different injections, representing when you harvest that mouse at time X, it would be 28 hours or 16 hours and 2 hours post-induction parasites in that one mouse. So using that, we can minimize the number of mice and cover the entire time course that we wanted to cover. So you then bleed all of those and do and pool and purify them, and we do single cell RNA-seq for this. So this is what the published data now looks like. So this is from the Matt Marty study, and you see the entire single cell range of blood stage parasites, including then the male and female lineage and the early gametocytes. And that's also seen slightly differently, where the focus was on different parts of the blood stage development, just showing and focusing here on gametocyte development. So these are the two areas that we would be most be interested in, remembering that what we're doing is inducing. So we're actually only looking at this section of the developmental cycle. And that's what it looks like. So when we look at the in vitro lines, then we expect to see males and females, and we see two very nice lineages bulbing out and giving what we would anticipate are going to be male and female gametocytes. However, in vivo, you see a completely different shape, which we would anticipate this larger mass of cells is going to represent the cells which have failed to mature as a result of carrying the acetyl-CoA point mutation. And then when we look at the sentinel genes, AP2G is expressed throughout. This is just a general early stage development, so you can see when AP2G is expressed. Then when we look at the male markers, you can see one lineage lighting up very quickly in the in vitro matured gametocytes. The female, it's the opposite arm. So we do seem to have authentic male and female gametocytes, and an asexual marker then is restricted to the bulb at the bottom, and these are presumably cells which have failed to respond to rapamycin and flip their cells. When we look at the parasites which are, excuse me, when the parasites which from in the in vivo maturation, you can see then that this arm here is the male arm. This very poor arm down here is the female arm, and the asexuals are here. So it looks as though the majority of the parasites are not maturing, and as we would have expected, and the majority of them are female. But it also would seem as though there are a lot of males which fail to mature as well, given the proportions that are here and the fact that we get more or less equal numbers of males and females when they're matured in vitro. So we have now a very great signature of what the arrest looks like, and we're going on to examine that in much greater detail. We'll be looking at metabolic signatures of this parasite population to try and understand what's going on and what the movement of acetyl-CoA means as an importance in terms of the signaling of the development of gametocytes. So we can happily say that these are the females and these are the males. Good representation in this. We've got two lineages here, and we wonder whether or not one is the cells that were destined to become gametocytes anyway, and then the other might be the cells which would normally become gametocytes on the next cycle, and that simply flooding the system with AP2G forces all of these cells to head off in the direction of gametocytogenesis. So to conclude, point mutations in the acetyl-CoA transporter give rise to ganoplacide resistance in malaria parasites. The ganoplacide-resistant acetyltransferase mutants grow slowly and cannot be transmitted. Female gametocytes are more severely affected, but both sexes are infertile. That might have some interesting consequences in terms of how you might deploy ganoplacide. So it's first of all, the only in vivo data concerning the imidazole piperazine resistance comes from the mouse and the mouse system and Plasmodium berghei. Plasmodium falciparum, in contrast, of course, is always kept in culture. So if you think about that, then the gametocytes in the resistant lines would mature, because that's what we see when we put our arrested gametocytes from the blood into culture. So there's something about the culture conditions, which of course we're interested to understand, that allows the arrested gametocytes to re-enter the developmental pathway and actually go through to full maturation. Having said that, the ganoplacide-resistant gametocytes are infertile, therefore the resistant form cannot be transmitted. And so that means it could be a perfect partner drug, because if resistance to ganoplacide is comes about, then that cannot be transmitted. And if using combination therapies, then if the resistance to the second drug arises, then ganaplacide should kill that. So it looks as though it's an extremely useful drug. So resistance to the partner drug will also not be transmitted. So I think that makes it a pretty good partner. We need to generate data in the field in order to be able to understand this. That's not going to be a straightforward proposition, and we're struggling now to find areas where we can try to establish this as fact. But this is something that we're interested in doing, and we can generate the mutants, of course, in the laboratory, and then we may have to look at human-controlled infections in order to be able to demonstrate that further. So I'll stop there. Thank the team in general. I've highlighted the people who've produced the work that I presented here, and of course, huge thanks to Thierry for his ongoing collaboration. Thank you very much. Happy to take questions.
Wonderful. Thank you, Andy. We had scheduled a very extensive period for question and discussion. I think we're going to have to cut down on that a little bit. But if anyone has some burning questions, I invite them to speak up now or write them in the chat. And I'm sure that if more questions will come later, you can also write them in the chat and you can answer them as we go. Stefan.
Many thanks, Andy. Fascinating. It was a lot of data. I'm still trying to wrap my head around what has been presented. So, in fact, sort of for the falciparum, of course, is the most relevant, interesting system for us and for using the drug in the field. I don't. Well, is this more kind of a speculation about the in vitro culture conditions, because they are in culture, because for instance in Nijmegen, Leiden and elsewhere they do transmit happily. So even though they are cultured, this is NF54. Sorry, I may have missed the whole point of why would falciparum, because of the culture conditions, respond differently. Would have a phenotype or...
Sorry, so my point is that in vivo, you see the arrest. You take the parasites out of from the in vivo setting, and then so these are arrested gametotypes, put them into culture, they re-enter development and achieve full maturity. So with falciparum, because it's already in culture, then you're never going to see that arrest. So my point was really that it's another value of the rodent model using an in vivo setting, is that phenotype was observed and couldn't in vivo, and would therefore have been missed and has been missed if you rely on falciparum data alone.
And how about using a humanized mouse system for falciparum, where you could study falciparum in vivo?
You could do that. You could do that. I just a little dubious about using such artificial systems, and I would much prefer to Dr. Takeo and colleagues and use the controlled human infection for that.
Yeah.
But one could do that, and I suspect Takeo would be perfectly within his rights to demand me to do that first.
Any further questions? So if not, I do have a question, because I was fascinated by the bifurcation that you saw within the single sex upon induction of the lines, and you already alluded to that. You think that might be a segregation between naturally committed and artificially committed parasites. If you separate those two data, those two groups, and look at them, like analyze them again after treating them differently, do you see anything that, well, do you see anything at all, anything that alludes to like slightly different transcripts, behavior, anything that would be able to tell them apart?
I'd have to pop next door and ask Jonathan, because that data we only discussed and generated and saw for the first time at the beginning of last week. So
all right. Well, then I'll repeat the question in a few
work
months.
in progress.
All right. Well, for the sake of time, let's move ahead, because we still have a second talk from Mariana. So thanks again, Andy.
Is Mariana for the delays?
Mariana is a postdoc in the lab of Sheiner. Again, I will keep the introduction very brief, so we have more time for the science. And she'll talk about my favorite organelle in Toxoplasma gondii, working on the redox regulation in the mitochondria. Mariana, stage is yours.
Thank you, Taco. Good afternoon, everyone. So today I will present my work it's about elucidating the role of a novel mitotyrodoxine in the redox regulation of Toxoplasma gondii. So first of all, what are thyrodoxines? I don't know if you might have heard about these enzymes before, but these thyrodoxines have a very specific domain that contains two active cysteines, and they are responsible to control and regulate cells in different organisms that are under stress. So basically, this thyrodoxine reduces its substrates and disrupts disulfide bonds from these specific substrates. In plants, it has been shown that thyrodoxines regulates both the TCA cycle and the mitochondrial electron transport chain. So upon stress conditions, thyrodoxines downregulates the SDH complex and upregulate the alternative oxidase in plants. Also, in chloroplasts, it was shown before that thyrodoxines have a mechanism to switch it off the ATP synthase upon light availability. So during the night, that there is a less electrochemical potential and less light availability. One of the ATP synthase subunities, the gamma subunity, has specific substrates that are in oxidized conditions. And the ATP synthase is not fully functional and is not produce ATP. So during the day, upon light availability, there is an electrochemical potential being generated, and these same substrates are now reduced. So the thyrodoxine disrupts the disulfide bonds between these sulfides. And then the ATP synthase can generate ATP in its maximum conditions. So these are just a few examples on how thyrodoxines can directly regulate mitochondrial complexes in other organisms. So our main question is, what is the role of mitochondrial thyrodoxines in apicomplex and parasites, more specifically in Toxoplasma gondii? We all know that the mitochondria of Toxo is very divergent compared to the mammals one. So here I have a few examples of complexes from the mitochondrial electron transport chain in Toxo that have much more different proteins in bigger sizes and with different roles in the parasite compared to humans. Also, the mitochondrial electron transport chain in Toxo lacks the complex 1, so it has only 3 complexes plus the ATP synthase. And you can see here highlighted in blue and red all the divergent proteins in the ATP synthase complex in Toxo. So thinking about that, we were trying to see how these mitochondrial thyrodoxines can regulate the divergent complexes from the mitochondrial electron transport chain in Toxo. So to start our work, first of all, we had to select potentially thyrodoxine proteins in Toxo. And for that, we use an apicoplast thyroidoxine that it was shown and published by our group before as one of thyroidoxine in toxo. So here we can see the predicted structure of this ATRX, and we use this structure to find other potentially thyroidoxines in toxo that have a similar fold as this apicoplast thyroidoxine. We use fold-seq to see these predictions, and we found 104 potentially thyroidoxine genes in toxo. So from these genes, we matched these sequences with a thyroidoxine profile that we built using other thyroidoxines in different organisms, as bacteria, yeast, and humans. So from this matched sequence, we found 26 thyroidoxines in T. gondii that has the thyroidoxine domain and that has this profile that matched with other thyroidoxines in well-known organisms. From these 26, we found 4 that are predicted to be in the mitochondria according to the LOPID database. So here we can see we did some phylogenetic analysis, and I highlighted the 4 mitochondrial thyroidoxines. We can see that these 4 the mitochondrial thyroidoxines are divergent from other organisms, and we can see that they match basically with the coccidian organisms. And from these 4 mitochondrial thyroidoxines, we selected 2 that were predicted to be essential for the parasite to continue our work. So the first one we call mitothyroidoxine 1 and the second mitothyroidoxine 2. We colocalized them with a mitochondrial marker, TOM40, and we validated that these thyroidoxines are actually in the mitochondria. We did a conditional knockdown using a TET system to see what is if these proteins are essential or not for the parasite growth. And we could see, we use this plaque assay to see if the parasite can actually invade the host cells and lyse the host cells. And we could see that upon knockdown of mitothyroidoxine 1, we can see a growth defect, different from the mitothyroidoxine 2. So we decided to continue only with mitothyroidoxine 1 for the work. So our next question was to see if the thyroidoxine domain was important for the parasite. So we mutated one of the cysteines from the thyroidoxine domain, and we knocked down the gene, and we added a second copy of the gene with the cysteine mutated. And we could see that this cysteine was important for parasite growth because upon depletion of this cysteine, the parasite had a growth defect. Moving on, we wanted to see specifically in which complexes of the mitothyroidoxine, the mitochondrial electron transport chain, was affected by knockdown of this protein. So we did a seahorse assay. I don't know if you guys are familiarized or not with this assay, where we basically uncouple the ATP synthase from the electron transport chain, and we evaluate the maximum oxygen consumption from the mitochondria. So we add ATC, we measure the basal oxygen consumption and the maximum oxygen consumption. And then we added a tobacco that actually disrupts the transport of electrons. And we could see that this thyroidoxine affects the parasite oxygen consumption by METC. And then we looked individually if each complex was affected or not. So we looked first on complex 2, where we basically use this enzymatic assay, where we evaluate if the complex is active, the electrons are transferred to DUB, and we use a redox dye, DCPIP. This dye is actually, if the complex is active, this dye is reduced, and we could measure the difference in the absorbance. We use a positive control. This is a specific complex 2 protein that we showed before that has a defect in complex 2 activity. So we could see that the assay is functional and working. And we could see that the thyroidoxine does not affect complex 2. So we could not see a defect in STH activity. We tagged a complex 3 protein to see if we knock down the thyroidoxine, if there is a defect in complex 3 assembly, and we could see a minor defect in complex 3 assembly. And we also performed complex 4 activity, where we ran the parasite lysates in native conditions, and we supplemented this gel with cytochrome C and DAB to see complex 4 activity. And we could not see a defect in complex 4. So far, we only saw a defect, minor defect in complex 3 assembly. So we decided to look at ATP synthase assembly. And we knocked down the mitothyroidoxine, and then we probed this with ATP synthase antibody. And we could see here that there is a defect in ATP synthase assembly, and we could see that upon knockdown, we could see a smaller band forming here that we believe it's the F1 subunity from the ATP synthase. We then performed an assay to evaluate ATP production. That basically we supplement the parasite with glucose and glutamine. Glucose is involved in both glycolysis and oxidative phosphorylase from ATP synthase. And glutamine is only, the ATP production is only involved, produced by ATP synthase. So we supplement the parasites with both substrates and oligomycin, that is our negative control. We could see that upon supplementation with glucose, there is no effect in ATP production, but with glutamine, it is. So we believe that this thyroidoxine is involved in both ATP synthase assembly defect and ATP production. ATP synthase disruption also leads to a defect in mitochondrial crystal formation. So we evaluated through EM images that there is less crystal upon this mitothyroidoxine knockdown. And then we decided to find specific substrate from this mitothyroidoxine. We searched in the ATP synthase structure from disulfides, and we found that all the disulfides were in the inner membrane space. We found 2 potentially substrates that contain these cysteines close to each other, and then we knocked down this mitothyroidoxine and tagged in a tagged background from these ATPTGs. We could not see any specific interaction between them, and then we decided to look in a more general interactions from MTRX. We performed a pull down with this thyroidoxine, and we sent to mass spec. We found some specific substrates, and most of them were in the mitochondria, and the main one was in the apicoplasm. And we found some substrates that are involved in ATP transport, ATP carriers, or substrates that have ATP binding domains. This work is still ongoing. Now I'm trying to see more closely what are the main interactors for this mitothyroidoxine, but we believe that our main hypothesis that this thyroidoxine is involved in ATP assembly and production is still valid. Thank you, everyone, for the attention, and all the Sheiner lab for the help.
Wonderful. Thank you, Mariana. This excellent talk. Are there any questions from the audience? I'll kick off with a question from my side then. Because I saw in your phylogenetic schedule that there's not a single copy in Plasmodium species. Was that correct?
Yes, it's correct. It's
And
very
so
toxic.
what would you expect? Is there no functionality there for a mitochondrial thyroid reducin, or is maybe one of the others responsible for the same activity?
Actually, in terms of mitochondrial thyroidopsin, I think it's very toxo specific, but there was other thyroidopsins that were more universal, and then it was we found them we could identify them in Plasmodium. So I think I don't know which specific function that would have in toxon that it doesn't have in Plasmodium, but yeah, we could see these differences.
Because are these candidate targets? Are those components that are also conserved in Plasmodium, or are these very toxo-specific ones?
They are very toxo-specific ones.
Okay.
Yes.
Any other questions? Probably a number of, well, actually, from the participants list, I can see that already quite a few people have left. So, if not, then I think we can close off this session for now. So thank you again, Mariana, and thanks, Andy, for your presentations. Thank you, everybody, for participating. And I'll just try to wish everybody joyous holiday season, and hope to see you again in February, as this is the next webinar meeting. In January, we won't have a webinar scheduled. And also I'd like to remind you to fill in the feedback form. So once again, thank you all for joining, and hope to see you next time. Bye-bye.
Thanks, Taco.
Bye.
Thanks, Mariana.